and the male along the side. And the outside of our box should be alleles, or what we're going to call gametes. Okay, so remember that gametes, or all of our allele variations, are going to be haploid. Okay, and so then when we make stuff inside the box, okay, these are going to be our fertilization events. So we're going to end up making all of the possible or potential offspring that mom and dad can make. And this is a really key thing to remember as we're moving on through this, is that this is not mom and dad will have every single one of these children, right? They're not going to have 4 children. They will not have exactly 50% gone babies, right? Remember that this is potentially what they will have. So they will have a 50% chance of having a blonde child every time they have a child, right? So this is something we have to keep thinking about, right? This is the pool of offspring that they will be having children from. Okay, so let's work on doing one of these together, right? So we can practice setting these up. All right, so let's look just at mom for a hot second. Is mom heterozygous or homozygous? Heterozygous, good, right? She has 2 different alleles, right? A big one and a little one. Okay. Does mom have round seeds or wrinkly seeds? Round, good. Right? Mom has 1 dominant allele and 1 recessive allele. Remember, when you have a dominant allele, that allele is loud and bullyish and covers up, okay, the sounds that the other alleles make, so we're only hearing what that dominant allele says. And that dominant allele is round seeds. All right, let's look at dad. Is dad heterozygous or homozygous? Homozygous, good, right? Both of his are the same. Is dad going to be round or wrinkly? Wrinkly. That's a word now. All right, so in addition to being homozygous, right, specifically he's homozygous recessive. Okay? Both of his alleles are recessive or little alleles. Remember, if we're going to hear the little allele, right, little alleles, right, or recessive alleles make small noises. Okay, so the only way that we hear those small noises is if they are both small, which they are in this case, so we can hear that. And the little alleles are telling us wrinkly seeds. Okay, so now we know what the parents are. Let's look at what kinds of gametes they can make. Okay, so remember when we're making gametes, meiosis tells us that gametes should be haploid. Remember, that was the goal of meiosis 2, to make haploid gametes. So we want to make sure we have haploid gametes here. All right, so I'm looking at mom. If I'm going to make a haploid gamete, I'm going to make an egg. Okay, I have the capacity. Okay, if I'm going to be haploid, I'm going to put 1 allele in it, right? I can have an egg that has a big P in it. That's one possible choice. Or I can have an egg. I got a little dramatic right there. With a little P in it. Okay, so those are haploid, right? They have 1 allele. Mom is normally diploid, right? Having 2 copies. But she can make more than 1 type of egg because she has 2 to begin with. We're going to make an egg for each of her alleles. Everybody see what I did there? Now we're going to do the same thing for Dad. We want to ask the question, what kind of gametes can Dad make? Okay, so I'm going to look at his alleles and say, okay, a little spermy. Ooh, that got weird. A little spermy. Okay. I can make a little sperm for his first little p. He's got 2. I'm going to go ahead and make 2, even though they're the same. He's way too small for myself. Okay. He's got 2 little p's, so I'm going to make him 2 sperm, each with only 1 little p, so they're haploid, right? Okay, now I want to go ahead and move my gametes onto my punnett square using the rules we established. Mom goes across the top. One egg goes over each box. Okay, same thing with Dad. Right, remember our rule was Dad goes down the side. I want to be consistent about how these are made. One spermy sperm in front of that box. One unreasonably sized spermy sperm in front of that box. Okay. This is one of your checkpoints. Everything that's in front of these boxes should be haploid. Okay, I should only have one of every allele. Okay, so I have P's here. I should only have one P above every box. This is a really good way to check it. Make sure we made good meiosis gametes. Any questions about what we've done so far? Okay, so there we are with our one allele above each. Okay, now the next thing we need to do is make our potential Beves. Okay, so we're going to go through and do fertilization events. Okay, so I'm going to take all of Mom's eggs and I'm going to bring her down into a box. I'm going to bring her down into a box. And then I'm going to take Dad's sperm and I'm going to bring him into the box. And I'm going to do that for all my boxes. I'm going to take an egg and a sperm. An egg and a sperm. Okay. Now, when I'm writing these in, I want to make sure that I write the capital letter or the big letter first. Right, so in this first box, you see I have a big P and a little p. And I want to make sure that I write the big P first. And remember, our dominant alleles are the loud bully alleles. They're going to make sure their voices are heard. They're going to cover up the voices of the other alleles. And since they're going to do that covering up, we want them written first, so they're the voice we see. So we're going to go ahead and translate all of these. These are both little. Taking a little and two littles again. This is another checkpoint. Once I've done fertilization, okay, we've come up with all of the potential Beves. If I have babies inside these boxes, okay, babies are organisms, okay, fully formed, right? These all should be diploid. And you can help yourself even further in that whatever kind of coding system we have, they should look more or less like mom and dad. And now remember from your dragons, okay, they don't have to have the exact same letter combinations, right? There can be some variation. But the letters that you have should be the same, right? Mom and dad had 2 P's. Baby should have 2 P's. Right? If mom and dad had 2 P's and 2 W's, baby should have 2 P's and 2 W's. Okay, whatever it is, if mom has it, baby should have it, right? You should look similar to the parents. Okay, so if something has went funky here, if you have a box with 1 P in it, or a box with 3 P's in it, okay? Take pause. This is a good place to stop and go back and check. Remember, we don't want babies with the wrong number of alleles. Okay, mutant babies, picked on in preschool. And we don't want that. So we want to go back and figure out what happened. Okay, did I make a weird gamete? Did I carry 2 eggs over somehow? We want to go back and figure out what happened, so then we can come back through and make sure we have good, healthy Beves. And I cannot honestly stress that enough. Okay, this is one of the biggest pitfalls students run into, is they don't stop and double check. Does the answer that I've made here make sense? It's really easy to make a small mistake that will make a large impact later on. Any questions about this? There's our fertilization, there's our diploid babies. Okay, now all that's left is to interpret what we've gotten. So the first thing I can look at is, what are the genotypes of my babies? So I might ask a question that says, what is the percent chance of a homozygous recessive baby? Really shouldn't write these. I think that's what we learned from this. What is my percent chance of getting a homozygous recessive baby? 50%. Good. Okay, so really all we're doing here is counting boxes, right? I have 1, 2, 3, 4. I'm very emotional about this. 4 boxes. Okay. Remember, homozygous recessive means same. Okay. And the sameness we see is that they are recessive, right? They're little. I have 1, 2 boxes that fulfill that requirement. Okay, so 2 out of 4 boxes answer this question, or 50%, right? one way that we can look at this. The other way we can look at this is by doing what's called a genotypic ratio, which is looking at the proportion or count of boxes we have for all of our different genotypes. Remember, genotypes are our allelic codes. Okay. So to do this, the first thing I'm going to do is list out the different genotypes I have. Okay, so there are my two genotypes, heterozygous and homozygous recessive. Okay. And now I'm just going to say how many boxes of each I have. Okay, I have 2 boxes, 1, 2, of the heterozygous. Okay, and I had, we already said, 2 boxes of that homozygous recessive. So I would report that like that. I could also say 1 to 1, right? Like a fraction, you can reduce that down when the numbers are the same. Does everybody see what I did there? Okay, so I'm just basically saying what's the proportion of options for a baby's genotype. All right, now I want to do the same thing for phenotype. Let's get rid of this. Okay, trouble is, just by looking at this, I don't actually have that information, right? What I have here are the codes. Phenotype, remember, is the physical characteristics. So the first thing we need to go through and do is actually translate the boxes that we have. Rude. What's box 1? Round. Good, right? We have that dominant allele, so we're going to get a dominant phenotype. Box 2. Wrinkly. Round. Wrinkly. Okay, now we can ask the same sort of questions. I could say, what percent of the babies will be round? And you would answer it in the same way. Okay, we're also going to do the same thing for our ratios. I'll put it off to the side so we have more space. So the first thing I'm going to do is list the phenotypes I have. In this case, I have round and wrinkly. And again, we'll count the boxes that we have for them. 2 and 2, or you can reduce that and say 1 to 1. Now you see in this example that our genotypic and phenotypic ratios happen to be the same. We're going to have plenty of examples where that is not the case, so don't assume on stock that that will always be so. They always count. Any questions about the example we've worked through together? Either how we set it up or how we derived our information from it when we were done. Okay, so what I want you to do then is in one of 3 ways, either from the email I sent you, it was posted on eLearn, or if you actually did manage to dig it out from the in-class activity, we have a whole worksheet that we're going to work through for our in-class activity. So I want you to pull that out or up or whatever is appropriate for the way that you do these things. And we're going to work through question 1 together. So you're going to actually take notes on this, right? And then you're going to turn this whole work packet in when we're done. So if you did not, and you should always be checking your emails, right? If you did not pay any attention to any of this stuff, do make sure that whatever notes you take in class today that you transpose onto that, be continuing forward on Monday. All right, so as we look at how to fill these out, you see we have several boxes that are going to help us make sure that we do all of these things as we go. So our legend is just simply asking us for what is our sort of our key, or what letters are we going to use. So if we think about our last example, that would just be the part that said big P is round seeds, little p is wrinkly seeds. Okay? So you're going to build your own. So what letters are you using? What's dominant and what's recessive, and what do they mean? Okay. The cross then is, what are the parents' genotypes? So on the previous example, remember we said mom was heterozygous, she was a big P, little p. And dad was homozygous recessive, he was 2 little p's. That's the information you would put in cross. What's mom? What's dad? Okay, gametes then are like what we did. If mom is big P, little p, okay, what are the haploid egg cells that you can make? Okay, so we made an egg with a big P in it and an egg with a little p in it. And same thing for dad. What are the haploid sperm cells that you can make based on what his genotype is? And then, of course, heading over to Punnett square, using those gametes, set up a Punnett square, and figure out what all of the potential Beves are. All right, so take a couple minutes and start working through this. So here we don't know that. Yes. So we don't know which one he is. So I don't know. Why don't we know? Because we're moms, right? Why don't we know? We've already seen it like this. I don't understand, right? Let's talk a little bit about the setup for this one, right? So and you might have some variation on this. Okay, so let's just be very clear. So if I were to make the legend, I might pick E as my letter. You do not have to use the letter E, right? You can use the letter A, you can use the letter Z. It does not matter. I'm just choosing the letter E because it has earlobes, but who cares, right? So I'm choosing E, right? A dominant and a recessive. If I go through the question, unattached earlobes are dominant. So E is unattached. Okay, that would imply, but we also do say that attached are recessive. Okay, so that's my legend. And don't feel the need to change your letters to match mine. Okay? Just make sure your dominant and your recessive is appropriate. That should be the same. Okay, so for the cross then, okay, I have a male and a female. Okay, so mom, she's right above me, we'll go ahead and look at her, has attached earlobes. Okay, I know if you're attached, you're recessive. Recessive is easy. Okay, remember, if you're going to have a recessive trait, they have to both be recessive. Okay, to hear that recessive voice, right, you have to both be really quiet. Okay, so I know that mom's going to have two, how nice those E's are, two little E's. Okay, dad has unattached earlobes, so we know his phenotype. Okay, so I know dad has to have at least one dominant, right, to get that unattached. What's dad's second allele? What'd you guys put? Why?
The dominant allele is the louder one as the boy or whatever. So even if we put the recessive allele, the second one, the dominant phenotype is shown on the page.
Absolutely correct. So she's pointed out that it can be either dominant or recessive, right? Since our first allele is the dominant one, it's going to bully or cover up whatever the second one is. So it makes it hard to actually know what it is without more information. So dad, kind of confusing. So we have two potential dads here. Oh my. So dad can be homozygous dominant or heterozygous. Right? Either of these would lead to having unattached earlobes. Which, if you're curious, just means you have a little space between your earlobe and your head. So some of them come straight down here, okay, kind of like at an angle, and some kind of come up in a loop, and you have a teeny bit of space. That's all we're talking about when we're talking about this. So those silly little facial features. All right. So if I'm making gametes, aligned between mom and dad, what kind of eggs can mom make? Recessive eggs, right? How many E's should each egg have? One. Remember, these are haploid. So I'm going to make two eggs, one for each E that she has. But she only has, am I whistling? She only has little E's to put in them. And now with dad, I would just pick one. We don't want to make this super complicated or huge. It's likely that you did when you did this. Okay? I want you to understand why there would be two choices here. We're not going to make this some enormous problem, right? This isn't statistics class. So, Dad, if you picked homozygous dominant, right, you're going to get two sperm that look the same, each with one big E in it. Or, if you chose heterozygous, you'll get a sperm with a big E and a sperm with a little e. Okay, so let's go ahead and set up the punnett square, although I'm not going to do it. Remember when we set these up, the key is mom on top, dad on the side. And then it's one gamete per box. So one egg over each box. And one sperm in front of each box. Right, and I'm going to leave my second sperm blank because that will depend on which dad you've chosen. Right, dad 1 or dad 2. Okay, so depending on what you've done will alter which offspring you get, and that will also alter your final genotypic and phenotypic ratios. Any question about John and his family of ears? So the one last thing we want to talk about is this question we have at the bottom: what type of inheritance? Okay, I'm asking what kind of allelic relationship do we have here? We've only talked about the first one, which makes it easier to narrow down. This is simple dominance. Okay, it's simple as in, right, a less complicated relationship, not meaning easy. Okay. In that case, it means we have one dominant and one recessive allele. Any questions about what we've done here? Okay, so make sure that yours is finished, of course. So as we think about inheritance types then, let's talk about what happens when you do, in fact, have a more complicated relationship. Okay, Facebook status, it's complicated. That's not at all what I want. This is what I want. So we're going to go through several of these. We'll introduce them, and then we'll go through examples of them in that packet that you have. So the first one we want to look at is called incomplete dominance. Okay, so the key here that we're noticing is that both genes or both alleles that we're looking at, okay, are dominant. So instead of having a dominant and a recessive, instead of having a loud and a quiet, right, both are loud. And this creates a very interesting environment for the resulting phenotype. Okay, so we have to imagine, perhaps, this is not so difficult, two individuals standing over the chip bowl, both yelling very loud and borderline incoherently about whatever it is that they feel very passionately about. As a result, absolutely nothing gets done, and you don't understand a single thing anybody says. That's not so hard to imagine. Okay. The key here then is the resulting phenotype. Okay, so what we get out of that mixed relationship is neither story. Right, so you've got one guy yelling very loudly about his trip to Africa, one guy yelling very loudly about his trip to Australia, and something kangaroos in South Africa. Like, you're pretty sure that's not right. You get this very strange blended story. Okay. So here's what we get. If we look at the example I have here, okay, our first allele, okay, is the R allele. Okay, we see this is a capital letter. We know this is dominant. I know, okay, and we can see this by looking at the image, that this allele codes for red flowers. And we can see this with the homozygous version, right? Homozygous dominant R version of this. We have nice, pretty red flowers. The second allele here is the W allele. This is also a capital letter, right? So we know this is also dominant. Here's where we get the yelling match. Okay. This allele codes for white flowers. Okay, so we can see when we're homozygous for W, which we have on the right side of the screen, we get pretty white flowers. Now, none of that is necessarily surprising. Even in our simple dominance, when you were homozygous for anything, you always saw that phenotype. So what makes this unique is if we focus on the heterozygote, where we have one of each. So remember, these are both still dominant. And here you can see the very clear blending. Okay, so you get a new, unique, and blended third phenotype. Okay, so this is really what's key here. Okay, we still only have two alleles. Just like with simple dominance, we only had two alleles there, right? Dominant and recessive. We only had two phenotypes, round and wrinkly, for example. Here we only have two alleles, big R and big W. And now we have three phenotypes, red, white, and then when they come together, they make a third intermediate phenotype, in this case, pink. This third phenotype is neither red nor white. I can't go in here and I can't circle any red bits. I can't go in here and I can't circle any white bits. Okay? This is wholly brand new. It is made a brand new, unique, blended phenotype. And this is iconic. This is special to incomplete dominance. This is one of the things you can use to tell it apart from some of the other complications we're going to look at.